Hello and welcome to BioRules video lessons. Today we are going to be talking about cells. We are going to be talking about the structure of cells, so how do they look like, especially how do they look like inside and how do they work. So the first type of cells that we're going to be working with are the prokaryotes. Prokaryotes are cells that have no nuclear envelope. They also have no kind of compartmentalization, so nothing to be separating the internal part of the cell. So basically, all the structures inside of a prokaryotic cells, they are not separated from each other by membranes. So they only have one membrane, which is the cell membrane that separates its inside to the outside of the cell. Here we have some examples of prokaryotes. So a few more pictures here for you. What are prokaryotes? Prokaryotes are also known as bacteria. So in this example here we have some Escherichia coli, E. coli. No, they are no bacteria, and so prokaryotes are all the bacteria that includes both eubacteria and archaeobacteria, which are two big domains of life. Independently from which of these domains we are talking about, they are going to be prokaryotes. Prokaryotes have roughly a very similar structure. So let's see how do they look like. First thing, when we are drawing a cell, try to make a clean drawing, something for us to be able to understand that you know what you're showing, that you can visualize things rather than having things messy that you don't manage to identify the structure. So first thing, draw an envelope, cell membrane. Usually structures in cells and in life, they are kind of roundish, so the arrows that we use to point for names, usually we try to make a straight line because straight lines are not very common in life. So that one is the cell membrane. Surrounding the cell membrane, actually embedded within the cell membrane, you have a cell wall. A cell wall is a structure that keeps the cell with a certain shape and avoids the cell from bursting. The cell wall from prokaryotes, they are glycoproteic. That means they are made of sugar and proteins in the same structure. The molecules that make those cell walls, they are sugar protein molecules. Outside of the glycoproteic cell wall, some bacteria may have a slime capsule. A slime capsule is a goo capsule around the cell. Let's take a look now inside. Prokaryotes, they have a nucleoid. It's not a nucleus, it's just a bunch of the DNA that get more or less together. And the DNA in the prokaryotes, they are naked. That means no nucleus and also no associated proteins. They are simply DNA. They are circular because they make a little ring. It goes and comes back. And that is, by definition, the genetic material, the DNA of prokaryotes. They have naked circular DNA. Additionally, prokaryotes might have plasmids, which are tiny little naked circular DNA. They are very small pieces of DNA that may also be floating out there. Okay. Prokaryotes, you have ribosomes. So the ribosomes here are 70S. 70S is an unit that makes reference to the size of the ribosome. So prokaryotes ribosomes are 70S, so they are smaller. Eukaryotes ribosomes, they are going to be 80S, that means that they are bigger. And if you are curious about the unit S, it's a unit of weight. And then if you are a very good physics student, you're going to come later to me and say, hey Pato, are you sure it's weight? Isn't it mass? So I can talk about that later. Okay. Additionally, prokaryotes, they have usually, or they might have pili. It's a bunch of fingers of membrane that protrude outside of the cell wall, and they usually cover all the surrounding of the cell. I'm showing here only one corner. It's basically the membrane that goes and makes a finger and comes back over here. Basically, it's part of the cell membrane, something like that. Prokaryotes might have flagellums. Flagellum usually is a good idea to draw it much bigger than the length of the cell. It can draw it 2, 3, 4 times bigger than the cell. Here, I made it smaller for it to fit, but it's a good idea to make it big. So prokaryotes may have flagellum. Eukaryotes might also have flagellum. You know spermatozoids, you know enterozoids, they also have flagellum. So this is something that we might have here. Something that some prokaryotes have, not all of them, is a mesosome. A mesosome is nothing more than the cell membrane that invaginates inwards, and you have here a projection of the membrane invaginated inward, making something like that. So basically it is cell membrane, but instead of protruding outside like a pili, it's folded inside of the cell. Okay? And of course, the liquid where everything is floating is called cytoplasm. So the cytoplasm is the place where chemical reactions are going to be happening. Also is the place where we have the DNA floating, the ribosome floating, the enzymes through chemical reactions, so they are all in there. If we look at an image of a prokaryote, it will look like this. This is an electron micrograph, and here it's quite clear where you have the cell wall, the membrane is in that border over there. We have the nucleoids over here. We might know these dots here. Many of them are going to be proteins and other stuff, but we also have ribosomes in there. But it's not very easy to see the other things. For example, we don't see plasmids here. We don't see the pili, we don't see the flagellum. Okay? Another drawing for you to see this is three-dimensional. When you're drawing cells, don't draw them three-dimensionally, it's quite difficult, but this one is very useful for you to see the pili surrounding the cell, like fingers of membrane protruding outside of the cell wall. Okay? They also have a flagellum there, quite beautiful. Okay? So this here is prokaryote cell, and now let's take a look on the functions of each of these things. So the membrane, when we saw the surface area to volume ratio class, you already learned that the membrane is responsible for what goes in and out of the cell. So basically it's what imports material, exports material, puts out the waste, so it's the in and out of the cell. Okay? What about the cytoplasm? I already told that in the previous class, and I told you again is where things are floating and also where you have chemical reactions. So basically it's a jelly-like structure, like a fluid, where you have the chemical reactions happening. Okay? The nucleoid is actually the DNA, so basically it's genetic information to make structures and material, things that the cells are going to be using. Ultimately, DNA is useful for the cell to make proteins. Okay? Ribosomes, they are protein factories. They are the place where the cell is going to be reading the genetic information to be making proteins for the cell to function. Okay? The plasmids are tiny little pieces of DNA and are usually for exchange. Prokaryotes, they don't have sex. They don't do meiosis. They divide simply by binary fission. They one cell blobs and becomes two, but they may exchange DNA through plasmids. And which is weird is that they don't only exchange DNA with similar bacteria of the same species, but they may even exchange plasmids with bacteria of totally different species. And that is very weird, very interesting, but very unfortunate for us, because sometimes a good bacteria may give some genes that we don't want a bad bacteria to have, and that can make the bad bacteria to be worse for our health. Okay? Pili, they are membrane projections, they are a place of contact with other cells, and they also increase surface area a little bit. Okay? Flagellum, of course, is related to movement. Sometimes not the movement of the cell itself, but the movement of the liquid surrounding the cell to make things to come to the bacteria itself, but they are related to movement. Okay? The cell wall, okay, it keeps the shape and it avoids the cell from bursting. Cells, they sometimes receive way too much material and they have a tendency of exploding. The cell wall don't allow the prokaryotes to explode. They keep them in their shape. Slime capsule, they are a protection, but they also take particles to the cell. They are a glue-like structure that stick things around. Okay? Mesosomes, they increase the surface area to volume ratio. Usually you're going to see mesosomes in very big bacteria, because as you saw in the previous class, when cells start growing, the surface area to volume ratio gets unfavorable. So bacteria that grows a little bit too much before they die or before they divide, they might work a little bit bigger if they have a little bit of mesosomes folded inside, which increase the surface area to volume ratio. Okay, so I hope you have enjoyed the class, and I will see you in the next one. Bye bye.